Usually when we talk about like a medical diagnosis or a biological process, there's this expectation of precision.
Right, like it's engineering or something.
Exactly. Like you break your arm, the x-ray shows that jagged white line,
Yeah.
and the doctor just points and says, there it is, broken.
Broken or not broken, very clean.
Yeah, it's clean. It's
Yeah.
binary. And I think it's comforting because we really like things to be visible, to be categorized neatly into a specific box.
We absolutely do. But then you step back and look at the macro picture.
Right. The absolute grandest scale of biology, the unbroken chain of living things on this planet. And suddenly, that X-ray machine feels a bit too small.
It really does, because when you look at that landscape, it is genuinely staggering. Every single organism on Earth, from, the mango tree in your backyard to the dog sleeping on your rug to you listening to this right now.
We all have a definite lifespan.
Yes, exactly. We are all born, we grow, we mature, we reproduce, and eventually we die.
And the only reason life still exists on Earth, like the only reason it isn't just some barren rock floating in space, is because of this biological baton pass.
An unbroken 4 billion-year winning streak.
Which is wild to think about. And that is exactly what we're exploring today on this deep dive.
We have a massive stack of biological research today.
Oh yeah, diagrams, classic experimental data, the works. And our mission today is to completely unpack how life continues.
From the very bottom to the very top.
Right, we are going to look at everything from the simplest single-celled organisms that essentially just, clone themselves, all the way to the incredibly complex genetic dance of human reproduction.
We are figuring out the how and the why behind the survival of literally every species you see around you.
Because the continuation of the species is, it's the ultimate biological imperative, right?
It is. A single mango tree might grow old and wither, but if it manages to scatter its seeds, new mango plants rise up.
The baton is passed.
Right. But what is truly fascinating here, when you look across all the domains of life, is that nature has essentially developed two completely different rule books for how to pass that baton.
There is a fundamental fork in the road of life, the big divide.
Exactly, the big divide.
Okay, let's lay out those two rule books for everyone. First, we have asexual reproduction.
Right, which is where a single parent produces offspring.
And those offspring are almost exact, identical copies of that parent.
Clones, essentially.
Great.
And on the other side of the divide, we have sexual reproduction.
Which is a whole different ballgame.
Totally. This involves two parents, and the offspring inherit a mix of characteristics from both individuals.
Which leads to variation and adaptation.
Precisely.
I like to think of this divide in terms of writing a document. Asexual reproduction is basically like hitting copy-paste on your keyboard.
Oh, that's a good way to look at it.
Right, you have one file, you highlight the whole thing, duplicate it, and you get an exact replica. Punctuation, typos, and all.
Nothing changes.
But sexual reproduction, on the other hand, is like two different people sitting down to co-write a brand new story from scratch.
They both bring their own ideas.
Yeah, their own vocabulary, their own style. And the final story is a totally unique blend of both authors.
That is a surprisingly elegant way to frame it, actually. That co-writing process leads to the small differences between parents and their young ones.
Which is super important.
It is the most important thing. Accumulated over many generations, those differences are the very mechanism that helps living beings adapt to changing environments.
And sometimes, over enough time, they even give rise to completely new kinds of species.
Exactly. But before we get to the complex co-writing process, we have to start with the copy-paste method.
Right, the ultimate biological cloning. Unicellular organisms, so things like bacteria, amoeba, and yeast, they rely heavily on this.
And simple multicellular organisms do it too, like the hydra or sea sponges.
But where it gets really wild, and frankly something you can actually see in your own house, is how plants pull this off.
Oh, plant cloning is a highly visible phenomenon.
Yeah, when plants sprout new shoots and roots from their existing parts, completely bypassing the need to produce seeds.
Which botanists call vegetative propagation.
Vegetative propagation. Let's define that clearly for the listener. It means the new plants are arising from the vegetative parts of a plant.
The stems, the roots, or the leaves.
Right. Rather than from a specialized reproductive seed.
Yeah.
And our sources give some brilliant everyday examples of this.
Fleshy underground stems are masters of this.
Yeah, like potatoes and ginger. If you are listening to this and you've ever, left a potato in the dark corner of your pantry for way too long, You know exactly what I'm talking about.
It starts looking a little scary.
It does. It starts growing those weird, pale little alien eyes all over it.
Well, those eyes are actually axillary buds.
Okay, what does that mean exactly?
So the potato is technically a modified stem designed to store starch. When you leave it in the pantry, the moisture in the air triggers those dormant buds.
They just wake up?
Basically, yeah. The cells inside those buds suddenly wake up and begin rapidly dividing. They are programmed to reach for light and establish a new plant.
Wow.
That is vegetative propagation in action.
Yeah.
You are literally watching a clone being born on your kitchen shelf.
That's crazy. And it's not just underground stems either. You can take a stem cutting from a money plant or a stalk of sugarcane, stick it in the ground, and it will grow into a completely new plant.
Yes, very common practice.
But the absolute wildest example we have in our source materials is the bryophyllum leaf.
Oh, the bryophyllum is
It
incredible.
looks like science fiction. Let me describe this for you because it's just bizarre. Imagine a wide, thick green leaf.
Like a succulent leaf.
Yeah. Now, all along the scallop serrated edges of this single leaf, tiny, perfect little miniature plants are just sprouting right out of the leaf tissue itself.
It's remarkable to see.
They have tiny green leaves reaching up and their own microscopic white root systems dangling down in the air.
It is an extraordinary adaptation. The bryophyllum has specialized cells along the margins of its leaves.
So just on the edges.
Right. And under the right conditions, these cells essentially forget that they are part of a leaf.
Wait, they forget.
They revert to a kind of generalized state. They divide rapidly and then differentiate into roots, stems, and leaves to form these plantlets.
No, they just build a whole new plant from a leaf edge.
Exactly. And eventually, those tiny clones get too heavy, they drop off the parent leaf onto the soil below, those little roots take hold, and they grow into independent plants.
Okay, wait, hold on. I need to push back on this strategy for a second.
Go for it.
If they are exact clones, I mean, if they have the exact same DNA as the parent leaf they fell off of, what happens if the environment changes?
That is the big question.
Or what happens if a disease hits? Are they all equally vulnerable? Like if a specific fungus mutates and attacks one bryophyllum plantlet and all the hundreds of surrounding plants are genetically identical copy-pastes?
Then they are all in trouble.
Yeah. Won't that single fungus just wipe out the entire population overnight? It seems like an evolutionary death wish to put all your eggs in one genetic basket.
Well, let's look at the mechanics of the alternative. You are identifying the major evolutionary risk of cloning, and you are absolutely right.
Okay, so it is a huge risk.
Oh, massive. Because there is no genetic variation, an asexual population is highly vulnerable to a sudden shift in climate, a new predator, or a novel pathogen.
Because nobody has a unique defense.
Right. If the genetic code of one clone lacks the defense mechanism to survive the new condition, none of them have it. The whole field dies.
Have we actually seen that happen?
Unfortunately, yes. We've actually seen this happen in human agriculture with cloned banana crops being wiped out by Panama disease.
Oh, wow. The banana thing. I've heard of that. So why would nature ever invent this? Why would any organism risk that level of vulnerability?
Because of the massive upside.
Which is?
Staggering efficiency. Asexual reproduction does not require the organism to spend precious time and energy finding a mate.
True. You don't have to go on a date.
Right, and it doesn't require the complex, energy-heavy biological machinery of creating specialized reproductive cells.
Or growing flowers, producing nectar to attract bees, all that expensive stuff.
Exactly. If you are a plant and you find yourself in a stable, highly favorable environment with plenty of sunlight and water, your best evolutionary move is to claim as much of that territory as possible, as fast as possible.
So you just multiply explosively.
It is a strategy of rapid colonization.
Okay, that makes sense. Fast, cheap, and highly effective. But, high risk if the weather changes or a bug shows up.
Very high risk.
But here's the thing. While that vulnerability is a huge risk in the wild, human beings looked at this natural cloning mechanism and realized we could completely exploit it.
We really did.
We figured out how to turn this biological copy-paste into an absolute superpower for our own survival.
This is exactly where biological theory intersects with human civilization. Scientists and early farmers observed this vegetative propagation and adapted it.
They didn't want to wait around for seeds.
No, because seeds might not grow true to the parent. You don't know exactly what you'll get.
Because seeds are sexual reproduction, right? So they are a mix.
Exactly. So they developed reliable, large-scale methods to force plants to clone themselves.
And our sources outline several of these agricultural hacks, and they are just brilliant in their simplicity. Let's walk through them, starting with the most basic one, cutting.
It's a classic for a reason.
So imagine you are a gardener. You have a beautiful rose bush, and you want 10 more just like it. You don't plant seeds.
No, you wait until the end of the plant's growing season and you physically cut off an overgrown branch.
But there is a specific anatomical requirement here, right?
Yeah.
You cannot just cut anywhere.
Correct. You have to ensure your cutting includes nodes and internodes.
Nodes are those slight swellings on the stem where leaves usually emerge, right?
Yes, those bumps. And they are crucial.
Right, because those nodes contain this specific, highly active plant tissue capable of shifting gears to grow new roots and shoots. If you cut a piece of stem with no nodes, it's basically just a dead stick, right?
It is. It will just rot in the soil. So you take your cutting with the nodes, then you strip the leaves from the lower half of the branch.
Now let's unpack why you do that. Because the sources specifically instruct us to strip the lower leaves. But if leaves are the solar panels of the plant, why rip them off when the plant needs energy the most to grow new roots?
It actually comes down to water management.
Water management, okay.
Leaves have tiny pores called stomata that release water vapor into the air. That process is called transpiration.
Like the plant is sweating.
Essentially, yes. When you cut the stem from the parent, you've severed its water supply. It has no roots to pull up more water.
Oh, I see.
If you leave all the foliage on, those leaves will continue to transpire, rapidly sucking whatever moisture is left in the stem right out into the air.
So the cutting will just dehydrate and die before it ever gets a chance to grow roots.
Exactly. By removing the lower leaves, you drastically reduce water loss. That gives the buried nodes time to detect the moisture in the soil and trigger root development.
Wow, so it's all about buying the plant time to perform its biological magic trick.
Yes, you are managing its resources for it.
So you take that stripped stem, you insert it into compost-rich soil, and the notes specify planting it at an angle of about 45 to 60 degrees.
Which exposes more of the stem to the soil.
You water it, and the nodes hidden underground push out roots, while the nodes above ground push out new leaves. Boom, you've cloned a rose bush.
And then human ingenuity takes it a step further with a technique called grafting.
Oh man, grafting. If cutting is a biological magic trick, grafting is straight up botanical surgery.
Botanical surgery, that is exactly what it looks like in the diagrams.
You are literally taking two completely different plants and forcing their circulatory systems to fuse together into one Frankenstein plant.
The mechanics of it are deeply fascinating. You start with plant A, which we call the stalk.
The stalk.
This is a plant already rooted firmly in the ground. You create a precise wound or slit in its stem.
Like a V shape or something.
Usually, yes. Then you take plant B, which is a twig from a different plant called the scion, and you carve the end of it to match the slit in the stalk.
So they fit together perfectly.
Right. You insert the scion directly into the wound of the stalk.
And the diagram shows the gardener wrapping that wound tightly with a cotton cloth or grafting tape. Why wrap it? To keep bugs out.
Partly to prevent pests and infections from entering the open wound, yes. But mechanically, the wrapping serves to press the inner tissues of the two plants tightly against each other.
What tissues are we talking about here?
Specifically, you are trying to align their cambium layers.
Cambia.
The cambium is a...
That is mind-blowing. The cells of two completely different organisms just decided to build a shared plumbing system.
It really is incredible.
But practically speaking, why do farmers go through all this trouble? Why not just use a cutting?
Because grafting allows you to combine the best genetic traits of two different varieties without having to breed them and wait decades for the results.
Give me an example of that.
Okay, imagine you have a lemon tree variety that produces the most delicious, giant, juicy lemons in the world. But its root system is weak, and it constantly dies from soil rot.
Okay, grapefruit, terrible roots.
On the other hand, you have a wild, bitter lemon tree whose roots are practically invincible. They can survive drought, disease, poor soil, anything.
Let's see where this is going.
You cut the trunk off the invincible tree, that's your rooted stock. You graft a branch from the delicious lemon tree onto it, that's your scion.
So the invincible roots pull up water and nutrients and feed them into the delicious branches.
Exactly. The resulting tree has the invincible roots of one clone and produces the perfect fruit of the other clone. It is the ultimate agricultural hack.
That is so smart. And if you don't want to do surgery, there's another method outlined in the source called layering. This one feels a bit more, I don't know, gentle.
Layering is very effective for certain plants.
You find a shrub with long, flexible branches. Again, lemon trees are apparently great for this. You take a branch, bend it down to the ground, and you just bury the middle of the branch under a mound of soil, leaving the leafy tip sticking out in the air.
The crucial difference here is that you do not sever the branch from the parent plant. Not yet.
Right, because it is still attached. The parent plant continues to pump water and nutrients into the buried branch.
But the darkness and moisture of the soil surrounding that buried section trigger the nodes in that specific area to stop making leaves and start making roots.
So it's feeding off the parent like an IV drip while it builds its own independent root system underground.
Precisely. And after a couple of weeks, once those new roots are strong, you just take some shears, snip the branch connecting it to the parent, and you have a brand new, fully established plant.
Just attached and ready to go.
Exactly. These methods, cutting, grafting, layering, they have been used for millennia. But our sources bring us into the modern era with a concept that fundamentally changed global agriculture.
Plant tissue culture.
Yes, plant tissue culture.
The diagram for this looks like something out of a sci-fi movie. Instead of a garden, you have these sterile glass jars lined up in a brightly lit laboratory.
Very clinical.
And inside each jar, sitting on a bed of what looks like clear gelatin, is a perfectly formed, vivid green miniature plantlet.
That gelatin is a highly specialized artificial nutrient medium.
So what's actually happening in those jars?
To perform tissue culture, scientists don't need a whole branch or a leaf. They take a microscopic scraping of cells from the very tip of a growing plant, specifically the apical meristem.
The apical meristem. Why that specific part?
This is a region where cells are dividing incredibly fast, and crucially, where viruses rarely manage to penetrate.
So you are starting with the cleanest, youngest, fastest growing cells the plant has.
Exactly. They place these cells in the sterile nutrient jelly, which is laced with specific plant hormones.
Oh, remember the bryophyllum leaf where cells forgot they were a leaf?
Yes, the exact same principle. The hormones in this jelly force these scraped cells to multiply into a disorganized mass called a callus.
A callus, okay?
Then they change the hormone mixture to trigger that callus to organize itself and sprout thousands of tiny, perfect roots and shoots.
And because they started with virus-free cells in a sterile lab, they end up with thousands of absolutely immaculate, disease-free clones.
Exactly. And the sources highlight how crucial this is for banana farmers, for instance.
Right, because bananas are massively vulnerable to disease because they don't have seeds. Every commercial banana is a clone.
And this brings us to the socioeconomic impact, which the notes heavily emphasize. There is an entire infrastructure dedicated to this.
Really? Like a government thing?
Yes. In India, for example, there are institutions like the Krishi Vigyan Kendras, or KVKs. These are agricultural extension centers operating under the Indian Council of Agricultural Research.
I gather you do.
Their entire mandate is to take this high-level biological science, like grafting techniques and tissue culture, and teach it directly to local farmers.
Oh, that's amazing. So it's not just academics in a lab. It's vocational training.
Exactly. They teach a farmer how to graft a high-yield scion onto disease-resistant stock. The farmer's crop survives a drought, their yield doubles, their income rises, and the regional food supply stabilizes.
We are talking about microscopic cellular biology, directly elevating the economic stability of human society.
It is a profound chain of cause and effect.
It really is. But we spend a lot of time on plants. Asexual reproduction is not limited to botany. We need to shift our focus down into the microscopic animal and fungal kingdoms.
Yes, we do. The sources dive into budding and spores next.
Let's start with yeast. Yeast is one of those things we use all the time to bake bread or brew beer, but we rarely think about what it actually is.
It's a single-celled fungus.
Right. And if you put yeast under a microscope in a sugar solution, you see these tiny oval-shaped cells. But if you watch closely, you'll see the cell wall at one end start to bulge out.
Like a little bubble.
Yeah. It pinches off into a smaller oval, like a bubble emerging from the side of a balloon.
That bulge is a bud. Inside the yeast cell, the nucleus has made an exact copy of its DNA and pushed that copy into the expanding bud.
And then what happens?
The bud grows, eventually detaches, and swims away as a genetically identical clone.
And it happens incredibly fast, right?
Very fast. Under optimal conditions, they replicate explosively.
But it's not just single cells that do this budding thing. There's a multicellular animal that does this too, called a hydra.
Hydra are remarkable creatures. They are simple, tubular animals that live in freshwater ponds. They have a ring of stinging tentacles at the top of their body.
The diagram in the text is wild. You have this green, alien-looking tube with tentacles, and sticking right out of its hip is a miniature version of itself.
Yes. In a hydra, asexual reproduction is essentially an act of cellular outgrowth. At a specific site on the side of the parent hydra's body, cells begin to divide repeatedly.
And it looks exactly like the parent, just a fraction of the size. It grows tentacles, it develops a mouth, and it stays attached to the parent, just swaying in the water current.
It continues to grow while attached, feeding off the parent's resources. Once it is fully mature, the base of the bud pinches off from the parent's body and the new hydra floats away.
It settles on a rock and begins an independent life. It's literally a macroscopic clone.
It really is nature's copy machine.
But let's talk about when this cloning process becomes a nuisance or, even a danger to humans.
Spores.
Spores. Imagine you leave a bowl of cooked rice or a slice of bread on the kitchen counter for a few days during warm, humid weather.
We've all done it.
You come back, and it's covered in these fuzzy black and green patches. Or you see black spots growing on a damp wall in the bathroom.
The historical context here is vital, actually. For centuries, human beings observed that phenomenon and genuinely believed in a concept called spontaneous generation.
Spontaneous generation, like it just appeared out of nowhere.
Yes, they thought that if you left dead matter in the right conditions, life just magically conjured itself out of thin air.
You can totally see why. I mean, you put a perfectly clean piece of bread on a table, lock the door, come back, and it's covered in a living forest of fuzz. To the naked eye, it looks exactly like magic. Where did it come from?
This was one of the great biological mysteries until the brilliant scientist Louis Pasteur entered the picture.
Ah, Pasteur. The sources dedicate a whole section to his paradigm-shifting work.
He designed an elegant experiment using swan neck flasks.
Swan neck flasks, how did that work?
He boiled meat broth inside these glass flasks to kill absolutely everything inside them. The flasks had long curved necks, shaped like an S or a swan's neck.
So air could get into the flask, but any dust or particles floating in the air would get trapped in the curves of the glass neck before ever reaching the broth.
Precisely. And the broth in those swan neck flasks remained perfectly clear and sterile indefinitely, even though it was technically exposed to the air.
But if he snapped the curved neck off, exposing the broth directly to dust falling from the air above.
The broth would turn cloudy and rot within days.
So he definitively proved that life wasn't magically generating from the broth. The life was already in the air, falling into the broth.
He established the germ theory. He proved that new life always comes from pre-existing life. The fuzz on your bread didn't spontaneously generate. It grew from invisible biological packages that were already floating in the kitchen.
And those invisible packages are spores.
Yes.
The notes describe, looking at this bread mold under a microscope, it's not just fuzz, it's actually a complex structure. You see these long, translucent thread-like roots called fungal hyphae. And they burrow down into the bread, secreting enzymes to literally digest your sandwich from the inside out and absorb the nutrients.
But the reproductive part is at the surface. Reaching up from those hyphae are vertical stalks. At the top of each stalk is a round, swollen sac called a sporangium.
It looks like a microscopic lollipop in the drawings. And inside that round sac, the fungus is doing the copy-paste routine. It is aggressively dividing its cells to create spores.
And when the sporangium matures, the sac bursts open. What is staggering here is the sheer mathematics of the dispersal.
How many are we talking?
A single sporangium can release thousands of spores. A single patch of mold can release millions. These spores are incredibly lightweight, usually single-celled, and they have tough, protective outer coats.
So they just float on the air currents in your house.
Yeah.
Every time you open a door or walk past, you stir up millions of them. They're virtually indestructible.
They just drift around the world, completely dormant, waiting.
And the second one of them lands on a surface that has the right combination of moisture, warmth, and nutrients. Like your slice of bread, the tough outer coat cracks open, a new hyphae thread shoots out, and the clone army begins multiplying all over again.
It is a ruthless and incredibly successful strategy for survival. But to truly understand all of this, the regenerating potato, the grafting of trees, the budding hydra, the millions of spores, we need to look at the cellular engine driving this entire copy-paste side of the divide.
Right, the biological mechanism making asexual reproduction possible, which is a process called mitosis.
Mitosis. Let's really drill down into how this works. What is the physical mechanism inside a cell that allows it to duplicate itself so perfectly?
Okay, so imagine a single cell. Deep inside its nucleus is its DNA. organized into distinct thread-like structures called chromosomes.
For an organism to clone itself perfectly, the new cell needs an exact, error-free copy of the parent cell's entire genetic instruction manual.
So the first step isn't dividing. The first step is actually doubling.
Exactly. Before the cell even thinks about splitting, it unzips its DNA and builds a perfect duplicate of every single chromosome. If the organism normally has, say, 10 chromosomes, it temporarily creates 20.
And then the protective wall of the nucleus dissolves. The chromosomes line up perfectly down the center equator of the cell.
Have you seen videos of this?
I have. Under powerful microscopes, the cell builds these microscopic cables, spindle fibers, that reach out from opposite ends of the cell, attach to the duplicated chromosomes in the center, and physically winch them apart.
They pull one copy to the left side and the identical copy to the right side.
It is a mechanical marvel. It really is. Once the chromosomes are separated at opposite poles, new nuclear walls form around each set.
Finally, the outer membrane of the entire cell pinches inward, eventually tearing the cell completely in half.
So you started with one parent cell, and you end with two daughter cells. And crucially, because of that precise winching process, each daughter cell has the exact same number of chromosomes and the exact same genetic code as the original parent cell.
No typos, no changes, pure replication. That is the engine of asexual reproduction. It's how the yeast buds. It's how the plant tissue culture grows. It's how you heal a cut on your own arm.
Yes, mitosis allows for explosive population growth and rapid tissue repair. But let's return to the philosophical problem we discussed with the bryophyllum leaf.
The vulnerability issue.
Right. Mitosis creates identical populations. In a world that is completely stable, that's fine. But the Earth is not stable. Ice ages come and go. Continents drift. Volcanoes erupt. Novel viruses emerge.
If every organism on Earth only reproduced through mitosis, the first global shift in climate or a highly contagious supervirus would have wiped out life on this planet billions of years ago.
Because there would be no variation to survive the change.
Precisely. Mitosis is great for volume, but to survive an unpredictable, hostile universe, nature needed an insurance policy.
It needed a way to constantly invent new genetic combinations, hoping that at least a few of those combinations would be equipped to survive whatever catastrophe happened next.
It needed a way to shuffle the deck.
And that brings us to the second half of the big divide, the co-writing of the story. Sexual reproduction.
The conceptual leap here is massive. Sexual reproduction abandons the idea of single-parent cloning. Instead, it involves two parents contributing genetic material to form a brand new, genetically unique individual.
But the sources immediately point out an enormous, seemingly impossible biological math problem here.
A doubling problem.
Let's think about this. Let's say we have two parent animals, and their cells each have 40 chromosomes. If they just smash their cells together to make a baby, that baby's cells will have 80 chromosomes.
And when that baby grows up and meets with another 80-chromosome individual, their offspring will have 160 chromosomes.
And then 320, and then 640. Within a few generations, the cells would be so bloated with DNA, they wouldn't be able to function. The nucleus would physically explode.
You cannot just mash two full sets of DNA together. The math doesn't work.
Right. The math doesn't work with mitosis. Therefore, the biological solution to sexual reproduction required the evolution of a completely different, specialized type of cell division.
This process is called meiosis.
Meiosis. This is the mechanism that prevents the DNA doubling catastrophe. Let's look at how it works, using humans as the example. Okay. Every single normal cell in your body, your skin cells, your heart cells, your liver cells, has a fixed number of chromosomes. For humans, that number is 46.
But those 46 chromosomes are not just a random jumble. They are organized into pairs. We have 23 pairs of chromosomes.
And this is the critical part. For every pair, you inherited one chromosome from your biological mother and the corresponding chromosome from your biological father.
The sources refer to a cell with the full 46 chromosomes, the 23 pairs, as being diploid, meaning double.
Diploid. Got it.
But when it's time to reproduce, the body has to create specialized cells that aren't double. It has to create cells that only carry half the genetic load.
These specialized cells are called gametes. In male animals, the gametes are sperm cells. In female animals, the gametes are egg cells.
And in plants, the terminology shifts slightly. Pollen grains carry the male gametes, and ovules carry the female gametes, but their function is identical.
So how does meiosis actually make a gamete?
Meiosis starts like mitosis. The cell duplicates its DNA, but then, instead of dividing once, the cell divides twice.
Oh, a double division.
During this intricate double division, the pairs of chromosomes are pulled apart. The resulting gamete cells do not get pairs. They get only one single chromosome from each of the 23 sets.
So the gamete ends up with exactly 23 single chromosomes. The sources call this a haploid cell, half the normal number.
And that solves the math problem. When a sperm carrying 23 chromosomes fertilizes an egg carrying 23 chromosomes, they fuse together. 23 plus 23 equals 46.
The resulting embryo has the perfect fixed diploid number of chromosomes required to be a healthy human being.
It is mathematically perfect.
So hold on, but let me ask a mechanical question here, because this is where the shuffling the deck analogy really matters. When meiosis pulls those pairs apart to make a sperm or an egg, how does it decide which 23 chromosomes go into the gamete?
That is the big question.
Right? Like, does a sperm get all the chromosomes that originally came from the man's mother, or all the ones from his father? Does it always deal the exact same hand of 23?
This is the most beautiful mechanism in biology. The sorting is completely mathematically random.
Random.
The source material provides a brilliant thought experiment to visualize this using colored beads. It's a great way to make this invisible cellular process tangible.
I love the bead experiment. Imagine you have a bag, and inside are pairs of colored beads. These beads represent the chromosome pairs, and their colors represent different genetic traits.
Let's look at pair 1. It's made of green beads. You have one light green bead, which carries the genetic code for blonde hair, and you have one dark green bead carrying the code for black hair.
That's chromosome pair number 1. Now imagine pair 2 is blue. The light blue bead carries the code for straight hair, and the dark blue bead carries the code for curly hair.
When the cell undergoes meiosis to create a gamete, the pairs must separate. The gamete is only allowed to take one green bead and one blue bead.
But which ones it takes is essentially a microscopic coin toss.
Exactly. A sperm cell might randomly get the light green bead for blonde hair and the dark blue bead for curly hair, but the very next sperm cell the body produces might get the dark green bead for black hair and the light blue bead for straight hair.
And that's just the variation created by sorting TWO pairs of beads. We have 23 pairs. I remember looking up the math on this once. If you calculate the number of possible ways 23 pairs of chromosomes can randomly sort themselves into a gamete, it's 2 to the power of 23.
Which is an enormous number.
That is over 8.4 million unique combinations.
8.4 million different genetic hands that a single human being can deal into a single sperm or a single egg. And that is before you factor in another process called crossing over, where the chromosomes actually swap chunks of DNA with each other before they separate, creating even more infinite variety.
So when one of those 8.4 million possible sperm fuses with one of the mother's 8.4 million possible eggs, the resulting child is a genetic lottery ticket that has never existed before in the history of the universe and will never exist again.
That is why you look different from your parents. That is why you look different from your siblings, even though you were created from the exact same two biological sources. Every fertilization is a completely novel roll of the dice.
But let's bring this back to the evolutionary scale. Why did nature go through the massive trouble of evolving this incredibly complex, energy-intensive process of meiosis, randomly sorting chromosomes and requiring two individuals to find each other?
Because that random mixing is the engine of survival. Variation means that within a population, there will be a vast spectrum of traits.
So when something bad happens,
When the environment changes or a new disease strikes, the entire population isn't wiped out like our cloned banana crops. Some individuals, just by random chance, will have inherited a combination of genes that allows them to survive the new threat.
the sources give two fantastic real-world examples of human adaptations born from this variation. First, high-altitude tolerance.
Yes, the altitude adaptation.
If you take a person whose ancestors lived at sea level and drop them in the Himalayas, they will struggle to breathe. Their cells will starve for oxygen.
But populations who have lived in the high Andes or the Tibetan Plateau for thousands of years have genetic variations that actually alter how their red blood cells bind to oxygen. They thrive in thin air.
The second example is lactose tolerance. For most of human history, the ability to produce the enzyme lactase, which digests the sugars in milk, turned off after infancy.
But as humans began herding and domesticating cattle, a random genetic variation emerged in some individuals that kept lactase production turned on into adulthood.
In times of famine, being able to drink cow's milk without getting violently ill was a massive survival advantage. Those individuals lived, reproduced, and passed that specific genetic variation on. Today, large portions of the global population can digest dairy.
Neither of those evolutionary leaps would have been possible if humans reproduced asexually by cloning. Mitosis builds the army. Meiosis evolves the army.
Wow. Mitosis builds the army. Meiosis evolves the army. Exactly. Now that we understand the deep evolutionary why of sexual reproduction, we need to examine the physical how. How do organisms actually manage to orchestrate the meeting of these haploid gametes?
It is a mechanical challenge.
And to explore that mechanical challenge, we're going to look at the most successful, most diverse group of plants on planet Earth, the flowering plants,
Flowering plants, scientifically known as angiosperms. The defining feature of an angiosperm is that it uses a flower as its primary reproductive organ.
which is a concept that often requires a shift in perspective. To humans, a flower is an aesthetic object. We put them in vases, we paint them, we breed them for their scent, we view them as decorations.
Right. The notes point out how funny this is. Roots are practical, they dig for water. Stems are structural, they hold the plant up. Leaves are industrial, they perform photosynthesis. And flowers. We just assume they exist to be pretty.
But the truth is, a flower is not a decoration. A flower is a highly engineered, aggressively functional reproductive factory.
To understand how that factory operates, we need to dismantle it. The sources ask us to conceptually dissect a typical flower to examine its architecture. Imagine slicing a flower right down the middle, a longitudinal section, so we can see all the layers, which the botanists call whorls.
Let's walk the listener through this dissection from the outside in. At the very bottom, forming the base of the flower where it connects to the stem, you have the sepals.
These are usually small, green, leaf-like structures. When the flower is still just a young, tightly closed bud, the sepals form that tough outer green wrapper that protects the delicate reproductive organs developing inside.
Once the bud opens, the sepals fold back, revealing the next layer inward. The petals. The petals are the colored, highly visible projections. This is the part we find attractive.
But we must ask the functional question. Why did the plant spend so much precious energy creating vibrant pigments, volatile fragrant oils, and sugary nectar? It didn't do it for human enjoyment.
So a flower is basically just a very expensive marketing campaign for plant genitalia.
That is a surprisingly accurate way to put it. The petals are the neon signs. The fragrance is the broadcast signal. The nectar is the payment.
Because the plant is stationary, it cannot walk over to another plant to mate.
It must hire a third-party courier, a bee, a butterfly, a bird, to carry its pollen. The petals are purely designed to attract and guide those specific couriers.
Okay, so the courier arrives at the neon sign. What are they interacting with? That brings us to the actual reproductive machinery in the center of the flower. First, we have the male organ, called the stamen.
If you look inside a lily, you'll see these long, thin stalks sticking up in a ring. The stalk itself is called the filament. Its only job is to elevate the machinery into the air.
And balanced at the very top of each filament is a swollen, usually yellow or orange pod called the anther. The anther is the manufacturing center.
Inside its chambers, cells are undergoing meiosis to produce thousands or millions of microscopic pollen grains. And inside each one of those tough little pollen grains is the male gamete, the sperm cell of the plant.
Surrounding the stamen, right dead in the absolute center of the flower, is the female organ, the pistil. The pistil is built like a vase with a long neck, and it's made of 3 distinct subparts.
At the very top, reaching up to catch the pollen, is the stigma.
The structure of the stigma is critical. The sources note it is often flat, providing a landing pad, and more importantly, it is sticky.
It secretes a viscous, sugary fluid. This isn't an accident. It acts like biological flypaper to trap any pollen grain that brushes against it.
Below that sticky landing pad is the style. This is the long, narrow neck of the vase. Its job is essentially to create distance and act as a conduit.
And at the very bottom of the style, deep inside the protective base of the flower, is the swollen chamber called the ovary.
The ovary is the vault. Inside this vault are tiny, rounded structures called ovules. And inside each ovule, waiting patiently, is the female gamete, the egg cell.
The sources suggest that if you take a flower and carefully slice the ovary open under a magnifying glass, you can actually see these tiny ovules lined up inside, looking like miniature pearls in a pod. It's incredible architecture.
But building the stamen and the ovary is only half the battle.
The fundamental problem remains. The male gamete is locked inside a pollen grain at the top of the anther, and the female gamete is locked inside an ovule deep inside the ovary. For life to continue, they must meet.
The transfer of the pollen grain to the stigma is the absolute prerequisite for everything else. This process is pollination.
The sources outline a classic scientific experiment, Activity 11.6, to prove beyond a shadow of a doubt how essential pollination is. It's an elegant setup using a common pea plant.
Imagine you have a pea plant with several flower buds that haven't opened yet. You divide them into 3 test groups.
In the first group, you take a bud and carefully tie a small bag made of muslin cloth over it. Muslin is a breathable fabric. It lets sunlight and air pass through so the flower can continue to grow and photosynthesize. But the weave is tight enough that no insect can land on it, and no pollen from the wind can blow onto it.
In the second group, you take a bud, gently pry it open, and use microscopic tweezers to surgically remove all the stamens, the male pollen factories. Then you tie the muslin bag over it. So this flower is completely isolated and has no male parts.
And the third group is your control. You just leave the flowers alone, totally uncovered, exposed to the bees and the wind.
The results are stark. The first group, bagged but intact. Sometimes they produce a small pea pod because the flower's own pollen managed to fall onto its own stigma inside the bag.
But the second group, the ones with the stamens removed and isolated in the bag, zero fruit. No pea pods ever form. They just wither and die because there is no pollen.
The control group, of course, produces healthy pea pods.
The conclusion is inescapable. The transfer of pollen is mandatory for the formation of fruit and seeds. But the experiment also highlights the 2 different pathways this transfer can take.
When pollen moves from the anther to the stigma of the same flower, or another flower on the exact same plant, it is called self-pollination.
But self-pollination, while reliable, doesn't achieve the ultimate goal of sexual reproduction, right? It's just mixing the plant's own genetics with itself. It doesn't create wide variation.
Exactly. It's an evolutionary fallback mechanism. What the plant truly desires is cross-pollination. This is when pollen is transferred from the anther of one plant to the stigma of a completely different plant of the same species. This guarantees new genetic combinations.
But since plants can't move, cross-pollination requires external agents, nature's couriers. And the strategies plants have evolved to utilize these couriers are fascinating.
Let's start with the oldest, most brute force courier, the wind. Plants like wheat, maize, rice, and many grasses rely on wind pollination. They don't waste energy growing colorful petals or sweet nectar because the wind doesn't care what a flower looks like.
Instead, they invest all their energy into aerodynamics and mass production. A wind-pollinated flower produces astronomical amounts of pollen, and the grains themselves are incredibly small, smooth, and lightweight, so they can catch the slightest breeze.
And their female anatomy adapts as well. The stigmas of wind-pollinated plants are often huge, long, and feathery. They hang outside the flower acting like massive biological nets, trying to trap any invisible pollen grain drifting by.
But the sources include a data table comparing the success rates of wind versus insect pollination, and it points out a harsh reality. Wind pollination is a terrible numbers game. It's highly inefficient.
It's like standing on top of a skyscraper and dropping 1,000,000 business cards into a hurricane, hoping one of them randomly lands on the desk of the exact client you want. 99.9% of that pollen just falls on the ground or blows into the ocean. It's a massive waste of biological material.
Which is why the evolutionary invention of insect pollination was such a game changer. Plants realized that instead of relying on random chance, they could enlist targeted couriers: bees, butterflies, moths, and beetles.
This is where the marketing campaign kicks in. The plant builds the bright petals and secretes the nectar. A bee, searching for food, lands on the flower to drink the nectar. While it's rummaging around inside the flower, it brushes against the anther.
And pollen designed for insects is mechanically different from wind pollen. It is usually larger, heavier, and covered in spikes, hooks, or a sticky coating. It physically clings to the tiny hairs on the bee's legs and body.
The bee finishes its meal, flies to the next flower of the same species, and as it climbs in for more nectar, that sticky pollen brushes off against the sticky stigma of the new flower. Delivery complete. It's the difference between dropping flyers out of a plane versus hiring a courier to hand-deliver a package to a specific address. The success rate per pollen grain skyrockets.
And it's not just insects. The sources mention birds like sunbirds or the Indian white-eye pollinating flowers like hibiscus and the coral tree. Some aquatic plants, like Vallisneria, use water currents to float pollen from one plant to another. But regardless of the courier, let's assume the delivery is successful. The pollen grain has landed on the correct stigma. What happens next?
This is where we move to fertilization, the mechanical journey that happens in the dark interior of the plant. Because the pollen grain is sitting on top of the pistil, but the egg is locked deep inside the ovary at the bottom. How do they meet? The diagram of this is incredible.
Once the pollen grain lands on the sticky, sugary surface of the stigma, It absorbs that moisture and chemically recognizes that it is on the correct species of plant. If it's a rose pollen grain on a rose stigma, it activates. The pollen grain literally
That
sprouts.
grows a microscopic tube, called a pollen tube, that physically drills its way down through the solid tissue of the style. It secretes enzymes that melt the plant tissue out of its way, creating a tunnel straight down toward the ovary.
It is a remarkable feat of cellular navigation. The tube is guided by chemical signals emitted by the ovule. Once the tube reaches the ovary, it penetrates the ovule. The male gamete travels down this tube, exits into the ovule, and finally fuses with the egg cell.
That singular moment of fusion is fertilization. The two haploid cells combine their 23 chromosomes, or whatever the number is for that plant, to create a single, fully diploid cell.
That newly formed cell is called a zygote. It is the absolute beginning of a new, genetically unique life. And the moment that zygote forms, a cascade of hormonal changes triggers the entire flower to transform.
The marketing campaign is over. The plant stops funding it. The petals wither and drop off. The sepals usually fall away. The stamens dry up. All the plant's energy is now redirected to the ovary.
The zygote inside the ovule begins to divide rapidly via mitosis, developing into an embryo, a tiny, dormant baby plant. The ovule itself develops a tough, protective outer coat, transforming into a seed.
And while the seeds are hardening, the ovary wall surrounding them begins to swell, thicken, and store sugars. The ovary transforms into the fruit. So when you eat an apple or a peach or a pea pod, you are eating the swollen, mature ovary of a plant.
And the purpose of that fruit is to solve the next major hurdle, seed dispersal. If an apple tree drops all its apples directly beneath its own branches, 100 seedlings will sprout in the shade of the parent. They will violently compete for the exact same sunlight, water, and soil nutrients. Most will die. The parent plant needs to send its children far away.
So it uses couriers again. It packs the seeds in a delicious, sugary fruit. A bear or a bird eats the fruit, walks 3 miles away, and excretes the seeds in a ready-made pile of fertilizer.
Or they use the wind again. The sources point to the dandelion and the madar seed. The dandelion seed is attached to a structure that acts exactly like a complex aerodynamic parachute. A gust of wind catches it, and it can travel for miles before settling into a new patch of soil to germinate.
It is a breathtakingly complex, interconnected system. And it's a system that humans, once again, learned to completely manipulate. The sources introduce a historical figure here who is crucial to this story, P. Maheshwari.
Professor Panchanan Maheshwari is globally recognized as the father of Indian embryology. His work in the mid-20th century revolutionized our understanding of plant reproduction.
The notes highlight that he was one of the pioneers of in vitro fertilization in flowering plants. Wait, IVF for plants? How does that work?
It is an incredibly delicate process. Maheshwari and his team figured out how to extract the unfertilized ovules and the pollen from plants. In a sterile laboratory environment using artificial nutrient media, they manually facilitated the growth of the pollen tube and the fusion of the gametes in a glass dish. They literally created test tube plant embryos.
Why is that a big deal? Why not just let the bees do it?
Because nature has built-in incompatibility barriers. A tomato pollen grain cannot fertilize a potato plant in the wild. The stigma won't let the pollen tube grow. But by doing it in vitro, Maheshwari could bypass these chemical locks. He could force the hybridization of species that would never cross in nature, exploring completely new genetic combinations. His 1950 textbook, An Introduction to the Embryology of Angiosperms, became the global standard for this science.
That deep understanding of embryology paved the way for modern agricultural science. The sources talk about artificial hybridization. This is where farmers don't use a lab, but they heavily control the physical environment.
It's a scaled-up, highly controlled version of the pea plant experiment. Agronomists will find a plant with a desirable trait, say wheat, that survives drought. They will manually remove its stamens so it can't self-pollinate. They bag the flower. Then they take pollen from another wheat plant that has a different desirable trait, say massive grain size, and manually dust it onto the stigma of the bagged flower.
They are acting as the bee, but with a highly specific genetic blueprint in mind. They are directing the evolution of the crop. And the notes mention that this has evolved into direct genetic engineering, where we bypass pollination entirely and use enzymes to directly insert the DNA sequence for disease resistance right into the plant's genome.
All of our modern food security rests on manipulating the mechanics of the stamen, the pistil, and the ovule. But plants, as complex as this is, have a distinct advantage. They can just release their pollen to the wind or bribe an insect to carry it. Animals face a completely different, much more visceral set of challenges.
Right. Let's cross over into the animal kingdom. Animals have to physically navigate the world to reproduce. The sources outline two universal challenges for all sexually reproducing animals. Number one, you have to ensure the male and female gametes actually meet in a chaotic world. And number two, you have to ensure the resulting young survive long enough to grow up and reproduce themselves.
How a species solves those two problems defines its entire evolutionary strategy. The text provides a fascinating comparison, highlighting a general trend in energy investment between different classes of animals, specifically contrasting aquatic animals like fish with terrestrial mammals.
Let's look at the fish strategy. The ocean is vast, and the currents are unpredictable. Many fish species rely on external fertilization. A female fish will swim over a reef and release thousands or even millions of eggs directly into the open water. A male fish will swim over the exact same spot a moment later and release a cloud of millions of sperm into the water.
The gametes meet and fuse entirely outside the body of the parents. It requires massive coordination of timing and location, often triggered by lunar cycles or water temperature.
But the survival rate is abysmal. You have millions of tiny zygotes floating unprotected in an ocean filled with filter feeders, predators, and harsh currents. 99.9% of them are eaten before they ever hatch. The parents provide zero protection. They just drop the gametes and leave.
It is a strategy of pure volume. You invest relatively little energy in each individual egg, but you produce millions of them, gambling that the sheer, overwhelming mathematics of the swarm will ensure at least two survive to replace the parents.
But then you look at mammals. Mammals operate on the exact opposite end of the spectrum. Mammals utilize internal fertilization. The male gamete is delivered directly inside the female's body, where it meets the egg in a highly controlled, protected environment.
And the investment is staggering. A female mammal produces very few eggs, often just one at a time, but the resulting embryo does not float away. It develops inside the mother's body, drawing nutrients directly from her bloodstream. She carries it, protecting it from predators and environmental shifts. Even after birth, mammals invest immense energy in nursing and rearing their young.
It's a fundamental shift in the game theory of survival. You either invest energy in creating millions of cheap gametes and abandon them to the harsh world, or you create a tiny handful of highly expensive gametes and invest all your energy into fiercely protecting them.
And that transition perfectly sets the stage for us to examine the ultimate high-investment reproductive strategy. The system that requires the most precise biological engineering, the longest gestation, and the most intense parental investment: human beings.
The final major section of our deep dive, human reproduction. This is where biology becomes incredibly intimate and staggeringly complex. The sources break this down meticulously, starting with the concept of reproductive maturity.
Unlike simpler organisms that can reproduce shortly after formation, human beings are born with an immature reproductive system. It lies dormant for over a decade. The text notes that the transition occurs during puberty.
Puberty is essentially the biological alarm clock ringing. The brain releases a cascade of hormones that triggers rapid physical and emotional changes. The reproductive organs physically grow and mature, and crucially, they begin the active process of meiosis to produce viable gametes.
The ultimate biological goal of this system is to facilitate the meeting of a sperm and an egg inside the female body, form a zygote, and nurture that zygote as it develops into a fully formed fetus inside the uterus. To understand how that gauntlet is run, we must first map the anatomy, starting with the male system.
The male reproductive system is essentially a highly specialized manufacturing and delivery network. The primary organs are a pair of oval-shaped structures called the testes. But anatomically, they are positioned in a very strange way. Unlike the heart or the liver, the testes are located completely outside the main abdominal cavity, suspended in a pouch of skin called the scrotum.
This external positioning seems vulnerable, but it is an absolute biological necessity. The testes are the factories where meiosis occurs to produce sperm, but the enzymes required for human sperm production are highly sensitive to heat. They cannot function at the normal core body temperature of 37 degrees Celsius.
So the body has to physically eject the factory from the hot core. The scrotum acts as a localized climate control system, keeping the testes a crucial 2 to 3 degrees cooler than the rest of the body. If they get too cold, the scrotum pulls them closer to the body heat. If they get too hot, it relaxes. It's a dynamic thermostat.
Inside those cooled testes, millions of sperm are manufactured every single day. But once produced, they must travel through a complex plumbing system. They move from the testes into a tightly coiled tube called the epididymis, where they mature, and then they are pushed up into a long, muscular tube called the vas deferens.
The vas deferens travels back up into the body cavity, loops over the bladder, and eventually joins with the urethra, which is the common passage out of the body for both urine and reproductive fluids. But sperm cells can't just travel dry. They need supplies for the journey.
This is where the accessory glands come in. As the sperm move through the vas deferens, glands like the seminal vesicles and the prostate glands secrete specific fluids into the tube, mixing with the sperm to create semen.
The sources explain that these fluids aren't just liquid volume. They are highly engineered payloads. The seminal vesicles add fructose, sugar, which acts as rocket fuel for the sperm to swim. The prostate adds an alkaline fluid designed to neutralize the naturally acidic environment of the female reproductive tract, which would otherwise kill the sperm instantly.
It is a complete life support package. And the sperm cells themselves are marvels of biological minimalism. The text describes their structure. A head containing the densely packed DNA payload, a midpiece packed with mitochondria to burn that fructose for energy, and a long whip-like tail called a flagellum that physically propels it forward.
I read an analogy once that is perfect for this. Let's compare the male gamete and the female gamete because the text emphasizes their extreme asymmetry. The human egg is immense. It's one of the largest cells in the body. It is packed with nutrients, cellular machinery, and the building blocks to start a new life. The egg is like a massive, fully stocked nuclear aircraft carrier. It doesn't move much. It just waits.
And the sperm.
The sperm is like a tiny, stripped-down speedboat. No cargo, no heavy machinery, no armor. Just a massive outboard motor and a single passenger carrying a USB drive of genetic data. Its only mission is speed and delivery.
That is an excellent visualization of the biological roles. Now let's examine the harbor where that delivery must take place, the female reproductive system.
Unlike the male system, the female reproductive system is almost entirely internal, nestled deep inside the pelvic cavity. It is built not just for gamete production, but for long-term incubation. It centers around a pair of almond-shaped organs called the ovaries.
The ovaries serve a dual function. They are the endocrine glands that release female sex hormones like estrogen and progesterone, which regulate the entire reproductive cycle. And of course, they're the repositories for the female gametes, the eggs.
And here is a massive difference from the male system. Males continuously manufacture millions of new sperm every day, from puberty until old age. But a female is born with all the eggs she will ever have. At birth, her ovaries already contain millions of microscopic immature eggs.
Most of those will degrade over time. By the time puberty hits, she has a finite reserve. From puberty onwards, the biological cycle begins. Roughly once a month, under the influence of hormones, a single, solitary egg matures and is released from one of the ovaries. This event is called ovulation.
Once that single egg is released, it doesn't just fall into the body cavity, it is swept into the open, funnel-like end of a tube. These are the oviducts, more commonly known as the fallopian tubes. They connect the ovaries to the central structure of the system.
The fallopian tubes are lined with... So we have the anatomy mapped. Now we must look at the event itself. How do the speedboat and the aircraft carrier actually meet?
It is one of the most brutal gauntlets in biology. During sexual intercourse, the male deposits semen containing roughly 300 million sperm into the vagina. From that moment, the clock is ticking, and the attrition rate is catastrophic.
The environment of the vagina is highly acidic to prevent bacterial infections. Millions of sperm die immediately despite the alkaline buffer from the prostate. The survivors must then navigate through the cervix, which is often plugged with thick mucus. Only the strongest swimmers make it through.
Once inside the cavernous uterus, they face the immune system. The female body detects them as foreign invaders, and white blood cells begin attacking and destroying thousands of them. The remaining sperm must swim up the walls of the uterus and find the tiny openings to the two fallopian tubes.
But the egg is only in one of the tubes, so half of the surviving sperm swim up the wrong tube and eventually die of exhaustion. The other half swim up the correct tube, fighting against the downward current of the cilia.
Out of the 300 million that started the journey, perhaps only a few hundred ever reach the egg. They surround the massive cell, their tails thrashing frantically. The heads of the sperm release special enzymes to literally digest through the thick, protective outer shield of the egg.
And then, the critical moment. One single sperm, just 1 out of 300 million, manages to breach the final membrane. The moment its head slips inside the egg reacts instantly. It releases a chemical wave that hardens its outer shell, turning it into an impenetrable fortress. No other sperm can enter.
The head of that winning sperm pops open, releasing its 23 chromosomes. They slide toward the center of the egg and physically weave together with the egg's 23 chromosomes. Fertilization is complete, the biological math is solved, You have a single diploid cell with 46 chromosomes, the zygote, a brand new genetic blueprint.
That zygote does not stay in the fallopian tube. It immediately begins undergoing our old friend, mitosis. It divides from one cell into two, then four, then eight, slowly rolling down the tube toward the uterus as a microscopic ball of cells.
Meanwhile, anticipating this exact event, the uterus has been preparing. Over the last couple of weeks, hormones have commanded the inner lining of the uterus to thicken dramatically, building up a rich, soft cushion of blood vessels and tissue.
When the ball of cells reaches the uterus, it burrows into this thick, bloody lining. This process is called implantation. Once implanted, the embryo taps into the mother's blood supply. It begins to develop a placenta to draw nutrients and oxygen. The woman is officially pregnant.
For the next 9 months, the mother's body acts as a life support system, redirecting massive amounts of energy to build a skeleton, a brain, and organs from that single blueprint.
It is a process so delicate that modern medicine has developed incredible interventions for when it fails. The sources touch on in vitro fertilization, IVF, for humans. Just as Maheshwari did with plants, doctors can administer hormones to a woman to extract multiple mature eggs. They extract sperm from a man. In the highly controlled environment of a laboratory, they combine them, observe the fertilization and the first mitotic divisions under a microscope, and then carefully physically implant the healthy embryo directly into the prepared uterus. We can bypass the deadly gauntlet of the fallopian tubes entirely.
But biology is built on cycles, and most of the time, fertilization does not occur. We have to talk about what happens when the speedboat never arrives. The egg matures, it travels down the tube, the uterus builds up that thick, expensive, blood-rich lining, and then nothing happens.
The human body cannot simply maintain that thickened uterine lining indefinitely. It is metabolically expensive. If the egg arrives in the uterus unfertilized, it simply degrades. The hormonal signals suddenly drop.
Without those hormones to sustain it, the thick lining of the uterus begins to break down. The blood vessels rupture, and the tissue detaches from the uterine wall.
This tissue, along with blood and the unfertilized egg, is expelled from the body through the cervix and the vagina. This is menstruation. The bleeding usually lasts a few days, roughly every 28 days. Once the lining is shed, the cycle resets, the ovary begins maturing a new egg, and the process begins all over again.
The biological clock keeps ticking. There is one final piece of this genetic puzzle that the sources highlight, and it's a profound one. We've talked about the 46 chromosomes. We know that the zygote gets 23 from the mother and 23 from the father, but what actually determines if that resulting baby develops into a biological male or a biological female?
This comes down to the 23rd pair of chromosomes. The first 22 pairs dictate things like eye color, height, and organ development, but the 23rd pair are specifically the sex chromosomes.
They come in two shapes, which scientists simply named after the letters they vaguely resemble: the X chromosome and the Y chromosome.
Biological females possess two X chromosomes in their 23rd pair, so their genotype is XX. Biological males possess one X chromosome and one Y chromosome. Their genotype is XY.
Now let's run the math of meiosis on this. When a female cell divides to create an egg, that XX pair splits. One X goes into one egg and the other X goes into another egg. Therefore, every single egg a woman ever produces carries an X chromosome. That is all she has to give.
But when a male cell divides to create sperm, his XY pair splits. Half of his sperm will receive the X chromosome, the other half will receive the Y chromosome.
This means the entire biological sex of the child is determined by which sperm wins the race. The mother's egg is sitting there with an X. If an X-carrying sperm fertilizes it, X plus X equals XX. The baby will be biologically female.
But if a Y-carrying sperm wins the race and fertilizes the egg, X plus Y equals XY. The baby will be biologically male. Because the sperm distribution is roughly 50-50, the statistical chance of having a boy or a girl is essentially a coin toss, entirely dictated by the male gamete.
It's an elegant, brutally random system, and the chapter concludes by noting that while nature designed this system for relentless continuation, human beings are the only species that actively choose to hit the pause button. We have developed birth control methods, condoms to physically block the sperm, hormonal pills to trick the ovaries into halting ovulation, and intrauterine devices like a copper T that alter the uterine environment to prevent implantation. We alone have decoupled the act of mating from the biological imperative of reproduction.
It is a testament to how deeply we have come to understand our own biology.
Okay, we need to take a breath and look back at the sheer scale of the journey we just took.
It has been a comprehensive overview of the fundamental engine of life.
We started by looking at the sheer ruthless efficiency of asexual reproduction, the copy-paste rulebook. We saw unicellular yeast budding in a sugar solution, and the hydrospawning miniature clones of itself in a pond. We explored vegetative propagation, how a forgotten potato sprouts eyes, or how a single bryophyllum leaf can drop dozens of identical offspring onto the soil.
We saw how human agriculture harnessed that cloning mechanism. The surgical precision of grafting a delicious lemon scion onto an invincible rootstock. The laboratory wizardry of tissue culture, scraping microscopic cells from a shoot tip to mass-produce virus-free bananas. And we unmasked the invisible floating army of fungal spores, driven by the error-free cellular copying engine of mitosis.
But then we crossed the big divide. We recognized the evolutionary danger of clones, and we explored sexual reproduction. We dove deep into meiosis, the cellular slot machine that halves our chromosomes and randomly deals out 8.4 million genetic combinations, ensuring that every generation is uniquely equipped to survive a changing world.
We conceptually dissected the flower, recognizing it not as a decoration, but as a reproductive factory. The petals marketing to the bees, the stamens producing the pollen, the sticky stigma catching it, and the pollen tube drilling down to the hidden ovule. We saw how this leads to seed formation, the evolution of fruit for dispersal, and how scientists like P. Maheshwari manipulated this with in vitro fertilization to revolutionize global agriculture.
And finally, we looked at the animal kingdom. The shift from the massive low-investment gamble of fish spawning millions of eggs into the ocean to the intense high-investment strategy of mammals. We mapped the human anatomy, the thermal regulation of the scrotum, the microscopic speedboat of the sperm, the massive resource-rich aircraft carrier of the egg. We walked through the brutal microscopic race through the fallopian tubes, the moment of fertilization, the burrowing of the embryo into the uterus, the shedding of menstruation, and the 50-50 genetic coin toss of sex determination.
It is a staggering amount of biological coordination happening mostly invisibly every single second of every single day, just to ensure that life on this planet doesn't stop.
It's overwhelming.
It is, and as we conclude, it raises a profound point for us to ponder. We started this deep dive talking about how reproduction is simply how life continues, a biological baton pass governed by nature, driven by random mutation and environmental pressure. But consider this. With our deep understanding of the mechanics we discussed today, tissue culture, artificial hybridization, genetic engineering, and human IVF, we have crossed the threshold. We are no longer just passive participants in reproduction, we are actively directing it. If natural selection relies on random variation in the harsh pressure of the environment, what happens to the future of life on Earth now that humans can artificially select, edit, and orchestrate the very building blocks of life? We are writing our own biological rulebook. The evolutionary consequences of that are entirely unknown.
That is a deeply heavy and absolutely fascinating thought to leave on. Thank you for joining us on this deep dive into the continuation of life. The next time you look at a dandelion seed blowing past you in the wind, or a fuzzy patch of mold on a piece of bread, or even just looking in the mirror at the specific shape of your own nose, we hope you see it all with a new sense of wonder. The medical X-ray machine might not show it cleanly, but the unbroken 4 billion-year chain of life is the most complex, brutal, and beautiful system in the universe. Keep questioning, keep exploring, and we'll catch you on the next deep dive.